John McLaughlin from San Jose State University. John, okay. Sorry, there's.
Here, if you just, there you go, you got it.
So John McLaughlin is the curator of bryophytes at the Charles Smith Herbarium at San Jose State University. John graduated from University of North Carolina in 2019 with a BS in biology and graduated from San Jose State University in 2023 with an MS in biological sciences. John's thesis, which he'll be discussing today, consisted of studying bryophyte flora of California's second largest state park, Henry W. Coe State Park. John, originally from New England, where his interest in natural history began as a hobby through foraging for fungi and ultimately moving to plants as undergraduate after taking a week-long workshop in the southern Appalachian Mountains of North Carolina and taught hepatologist Paul Davison. John focused his efforts on bryophytes. So we have John here, and we're really excited because this is actually a follow-up of last year's bryophyte talk. And so it's going a little bit more in depth. And so kind of the theme of this year's presentations was starting at the macro level or the micro level and going into the macro level. And so we're excited to have John speak. And so John, you can take over the screen. Let's see.
Stop share on the.
Yeah.
Yeah, I can go ahead and do it, and then it says it'll stop sharing your screen. I think. Yeah, let's try that.
There you go.
Let's try this, and then I'll put the presentation. All right, how does that look?
It looks good. Just give us 1 second. We just need to switch this.
Yeah, let's see here. Let's duplicate, maybe.
Yeah.
We might get this.
Perfect.
Then we have to get rid of all of it.
Standard.
I think we can this small. Yeah.
Okay. Go ahead, John, and we're gonna just
All right.
Here.
Perfect. All right, so yeah, if everything looks good, I guess I'll get started. Well, thank you so much for having me. It's a pleasure. I know my colleague, Jason Brooks, gave a presentation last year and very kindly recommended me. But Jason and I, we meet every year or so, discuss bryophytes, as there aren't many people who study bryophytes, especially floristics. And so it was good that Jason recommended me. Like Willard said, I'm a recent graduate in my master's program where I studied bryophytes, and now I'm working at Stanford University in the conservation program there, where sadly I don't get to look at bryophytes every day, if that often at all. But when I have time, I still like to work on research, studying bryophytes, and certainly going out and collecting and identifying bryophytes in my own time. So today I'll be showing you some research. Let's hop into it. But essentially, we're looking at current research in California bryology, things that I'm involved in, and to kind of span a spectrum here. So there you go. So you got that information. Okay, perfect. So I'll be covering 3 topics today under the context of these 3 studies that I'm involved in. So the first topic is a biogeographical topic, and it's talking about California bryophyte bioregions. The second topic is a floristic topic. So this is the one I did for my thesis, and it's the bryophyte flora of Henry W. Coe State Park. And then lastly, I'll be talking about speciation, or taxonomy-related study, looking at the morphological evidence supporting Anicolia baueri as a species. So I'm not sure out of the crowd we have here today, how many of us are, bryologists, vascular plant people, mycologists. Seems we have maybe have a mix, but I'll give a quick introduction to the group of plants that we'll be talking about today. So bryophytes include three main plant groups: your mosses, liverworts, and hornworts. And we can see them pictures of them here. So these are typically small plants, and when we compare them to other plants, we usually call them non-vascular plants. And so this means they don't have true vascular tissue as is defined in the angiosperm or higher plant world. Some other things about bryophytes that are important and distinct are, first of all, that they're ancient plants. So before all of our flowering plants, bryophytes are thought to be the first to evolve to live on land, evolving from algae. I think that was, roughly around 420 million years ago, they evolved to live on land. And because of this, they have some kind of some more basal strategies in terms of reproduction and morphology. So they reproduce via spores and have a dominant haploid gametophyte generation. Bryophytes have no true roots. So they have these things called rhizoids that keep them pressed and attached to their substrate. But beyond that, they obtain their nutrients and water through the atmosphere and through their leaves. And bryophytes are important for many reasons. So first of all, they are the second most diverse plant group out of all the plant groups we have today. So we have angiosperms and then bryophytes in terms of diversity. Yet they are significantly understudied. So other important things that they contribute to the ecosystems include nutrient cycling. Certainly carbon sequestering is a big topic with bryophytes that many people are familiar with when it involves peat moss or sphagnum, as you can see in this image here. Also, bryophytes are really important for a number of different animals. So a lot of invertebrates find themselves using bryophytes, having their whole life stages within bryophytes in terms of mating, using bryophytes for shelter. Birds use mosses and other bryophytes to make their nests. And so, yeah, lots of microscopic and macroorganisms will use bryophytes to their benefit. And another reason is ecosystem development. So in scenarios where the ecosystem is either disturbed, destroyed, or there's a fire, bryophytes and other things like lichens play an integral role in reviving that ecosystem and providing biomass, bringing the nutrients back into the ecosystem so that it can recover over time. Very good. So that's your little introduction on bryophytes. Next, we'll hop into the first topic, the biogeography. So this project here, California Bryophyte Bioregions, I first presented this last fall at the CNPS symposium or conference that was held here in San Jose. So this is a collaboration between me and my advisor at the time, Ben Carter, but it's all part of a larger study that's still being worked on, including other colleagues like Brent Micheler and a few other people who are doing some more advanced analyses that I won't be talking about today. So this little section of the larger study is just myself and Ben that worked on it. Okay, so a little bit of background for this study is the flora of California is some of the most diverse in North America. And bryophytes make around 12% of this diversity. Yet, looking at spatial patterns of bryophytes is something that hasn't really been done. So one study that included bryophytes in their spatial analysis was this study from Verge et al. 2016, and we can see that figure here. However, for this study, they did just the bare minimum for bryophytes, and they confined their results to looking only at the California floristic provinces. And if you're unfamiliar with the California floristic provinces, I'll talk a little bit about a little bit more about it later. But there are these ecoregions that the Jepson has created, and they kind of define these areas in California. And They also extend into Oregon and into Nevada and to other places, other bordering states and countries. So this study from Burgett et al. 2016 resulted in, like I said, a baseline understanding of bryophyte endemism and richness within California. So moving on to Baldwin et al. In 2017, looking at this figure here. This study looked only at vascular plants, and Baldwin et al. Basically tried to test the relationship between the California vascular flora and these ecoregions that I just mentioned. So shown here, California, from these two studies alone, and many others, California has acted as a model for studying plant biogeography. Yet, sadly to say, that bryophytes are rarely, if ever, included in these analyses, despite making up 12% of the diversity in the state. So basically what the goal of this study is to make this figure that Baldwin et al. 2017 did, make this figure but for bryophytes. And we're going to compare these bioregions, as we call them, bryophyte bioregions, with the Jepson ecoregions. And we're going to test, we tested 3 different data sets. So one has a high taxonomic resolution. So this data set is information that was only extracted from flora or floristic studies. So like the study I'll talk about later. So the sample size is lower, but in terms of taxonomic resolution, it's certainly high. Then we'll look at a data set that is confined by counties. And then lastly, we'll look at a data set that's confined by 50 kilometer square grid cells, and so has a high spatial resolution. And lastly, also we're going to talk about creating a collecting intensity map in California for bryophytes, because as you'll see shortly, that this data set is certainly incomplete, and there are many gaps in our knowledge of bryophytes in California and in the West as a whole. And so we can take a look at that as well. So our questions are: do the bioregions align with the Jepson ecoregions? Is one of those 3 maps better than the other at showing this? And then what differentiates in terms of species and family relationships, differentiates the regions that we'll define? And then again, what does all this tell us about our gaps in knowledge currently with understanding bryophytes in California? So our data comes in part from the Consortium of North American Bryophyte Herbaria, which is now the Consortium of Bryophyte Herbaria. They got rid of the North America and included everyone, I suppose. And so this data set included 803 species from California. And that is, depending on who you talk to, that's somewhere in the middle of what people believe is the total number of species in the state. And then in this data set, we have 50,336 unique occurrences. And so with all this data, we create basically a spatial matrix. So we have our coordinates tied to each record of a collection of a correctly identified bryophyte within California. And then so some of the methods, not so important necessarily for your purposes, but so we have this data set I just talked about, and then of course we have another data set, another spatial matrix that was extracted specifically from these published studies that studied a specific study area, or whether it's a national park or state park. That's another data set. And then we did some analyses, and we created these UPGMA dendrograms. So basically it looks at the spatial similarity of species compositions within California. And so we make these through a software called Biodiverse, as well as using the software R. And then we use this other software, Figtree, to design these figures and make them look pretty. Okay, so to the results. So these are the 3 dendrograms that we created, okay? And so we designated these clusters. So we can see there's different colors here. So there's one color here. These different clusters are designated via the branch distances and in terms of spatial clustering. So are these locations nearby one another? And you can see there's some decent clustering, and as well as there's some gray areas and some black areas. And those are the areas of the state that don't have enough data for us to confidently put them in one group or another. But all in all, the flora has 8 clusters, the counties have 8, and the grid cells has 6 clusters. And here are those dendrograms displayed or projected on their respective maps of California. And then to the right here is the Jepson ecoregions that we'll be kind of comparing these results with. So we'll go one by one and discuss the pros and cons of these results. So the flora's data, and here's some zoomed in on some of these dots and these points. So as you can see, like I said earlier, the spatial resolution is quite poor because we don't have published floristic studies for everywhere in the state. So we included some studies from outside the state in Arizona and in the Baja, Mexico. When looking at patterns arising here, we don't see too much of a pattern, and that just has to do with the sample size. The only pattern we do see is the historical studies, interestingly enough, all kind of grouped together in this kind of purple color. And so these are studies done in the 50s and the 70s and from Baja and from Southern California. But it just goes to show you that, yeah, we need to do one thing, publish more floristic studies. This, like I said, it has a high taxonomic resolution. We know exactly what species are in this area, as well as just collecting it generally. So not the best result out of all the ones we'll see today, but it certainly can be added to, and hopefully in the future we'll have more data, more studies to extrapolate from, and we can maybe get some more interesting results with this type of analysis. So looking at the counties figure this time. The counties is getting better, certainly. And here we have it compared to the ecoregions, and we can see some similarities. So we have different groupings here. We have kind of a Great Basin in the top right corner of the map in this kind of mustard yellow. We have 2 different desert regions, the purple and the yellow here. We have the Sierra Nevadas into the Cascade Ranges in this teal color. We have this kind of navy blue, which represents southern coast of California and the interior coastal ranges of California. You have the central coast in green, and then the Pacific Northwest kind of area in dark green. As you can see, there's still many gaps. So the white counties are the areas that we just didn't have enough records from to put them into, to confidently put them into one group or another. And here we have another interesting one, is this light blue here, and this is 2 counties. I can't remember their names off the top of my head, but this is definitely something to be looked into further. If you look back on the tree itself, you see this light blue one. It's actually more closely related, or the species composition in these 2 counties, Amador and Calaveras, are more similar to that of Inyo and Mono, and these kind of Great Basin floristic regions. So that's quite interesting. They kind of pop out from the Sierra Nevadas that's found here in the teal. Interesting. So we're getting better resolution. And then we move to this last figure here, and we get the best resolution. And so we can see, yeah, we have 6 different clusters here, 6 different ecoregions of bryophyte regions, some similar patterns. But in terms of the Jepson ecoregions, the bryophyte ecoregions are more broad. So the deserts are combined in the bryophyte regions. Again, the Cascade Ranges doesn't really pop out. It kind of all groups together with the Sierra Nevadas, which is interesting. But yes, we have the desert region. We have the Great Basin in yellow. We have in teal the Sierra Nevadas. In the blue here, we have the southern coast ranges and also the interior coastal ranges of California. In our light green, we have the central coast ranges and the Sierra foothills. And then in dark blue, we have this kind of Pacific Northwest region. And so now I kind of want to break down each region that we have identified and talk about what differentiates them in terms of species richness, endemism, and looking at the proportion or relative number of species per family in each region. So looking at the first region, the desert region in purple here, has the least amount of species, but they are unique species. And so we have some endemic species here, including this one shown, Pterygonerum subsessile. And this region largely consists of mosses from the families Podiaceae, Grimiaceae, and Bryaceae. Next, moving to this Great Basin region here in yellow. There are 257 species in this region, including one state endemic. It may occur elsewhere in the country, but certainly in California, it only occurs here, is Funaria flavicans. And here, in terms of the family distribution of species composition, there's a broader spectrum of groups represented here. So Grimiaceae, Podiaceae, Bryaceae, Brachytheciaceae, all kind of the same abundance, relative abundance in this Great Basin area. Next, we look at the southern coast range, or the southern coast of California and the interior coastal ranges in blue. And again, we have a increase in species richness. We have at least one endemic species, Pluridium mexicanum, that's not found in any other region. And this area differs from the Great Basin in having a less proportion of Grimiaceae and a higher proportion of Podiaceae and Bryaceae. Let's see, those are the first three. Now the last three here, starting with the Sierra Nevada and Cascade ranges. So this is the region that has the highest species richness, with 607 species represented in this region. So instead of using pie charts for this area, it's a little more helpful to look at just general trends within the data set. So of course, the higher richness pulls this region out in part. There's definitely a high richness of liverworts here and a high richness of sphagnum species here relative to the other regions, including endemic species like Nardia geocyphus. Next, moving to the PNW region, which I imagine would be most similar to some of this mosses and bryophytes you'd see in Oregon, at least in the western side. So in this region specifically, there's a number of dicranum species that are absent in all the other regions that are only found in this area, and also moving into Oregon and Washington. And so one of those is this dicranum boonjunii. I'm not sure how to pronounce it, but you got it. As well as there's a high number of sphagnum species here as well. But overall, compared to the Sierra Nevadas and Cascade ranges, this has a lower richness with 444 species. And lastly, the Central Coast and Foothills ecoregion here has 552 species with a particularly high amount of richness in the Gemmabrium genus, Tortula genus, with a very low richness of liverworts and sphagnum species. Although there are no endemic species to this region, here's just a picture of one of the common Gemmabriums you can find in this region. It's also interesting to note that this figure has given us, has shown some disjuncts within California. So the first one I want to talk about is this Great Basin disjunct in yellow here. And so these 2 points pop up along, I believe it's the San Jacinto Mountains. I think it's a San Bernardino National Area. And this is this range that occurs in Southern California. So this area maybe strikes a few questions, and one could ask themselves, does the vascular flora also have this disjunct? Is there a common pattern there? And then there's also this white dot right next to this other yellow disjunct that, it just shows us that we should definitely go in there and maybe take a closer look at this area and see if it truly is kind of an island effect of this species distribution within California. And the other disjunct I want to talk about is this purple spot here. This is a desert ecoregion in purple, and so most of the purple is clearly in the desert area of California in the southeastern part of the state. But we have this one area here, and this is geographically actually Carrizo Plain. And if you're unfamiliar with this is a really diverse area for both vascular plants and bryophytes, but it is very much a desert habitat. And so one could imagine that if these areas above this purple spot here, north of Carrizo Plain in the San Joaquin Valley, If more collecting would be done here, would this purple expand? And would the San Joaquin Valley just be another kind of desert region in the eyes of bryophytes? So now kind of closing in on this topic. So some future areas of research. Well, of course, like I said earlier, we have some collecting priorities that need to be established. So looking at this figure to the right here, this map, this is the collecting intensity of bryophytes within the state. The greener the square, that means the more bryophytes were collected there, and then white means very few bryophytes were collected. And so these, largely follow what we've been seeing in the previous figures. So certainly, in terms of spatially, we need to collect more in the San Joaquin Valley. That's this area here in California. The transverse ranges, which is over here. And then east of the Sierra Nevadas and the kind of Great Basin region also has lots of gaps in it. And specifically in terms of counties, this county down at the bottom of California, bottom right, Imperial County, has I think maybe 20 collections in it. So it's really, really poorly understood. Although it is largely desert, there are more bryophytes to be collected there. In terms of collecting, prioritizing certain groups of bryophytes, if we look at this figure here, the bar chart, we look at the proportion of collected species by the proportion of species in California by phylum. So if we look at bryophytes first, so Bryophyta is bryophytes, Marchantiophyta is liverworts, Anthocerophyta is hornworts. For the bryophytes, the numbers, the percent collected versus the percent of species is well represented. When we move to the liverworts and the hornworts at the bottom, we see that the percent collected doesn't really match the number of species. And so we can say that the bryophytes are fairly represented in terms of collections, in terms of vouchers, but Marchantiophyta, the liverworts, and Anthocerophyta, the hornworts, are poorly represented. And so these, certain bryologists only collect mosses, certain bryologists like to focus on liverworts and hornworts. But if you're doing floristic work and you're collecting, you should collect all of them, because why not? And so some major takeaways here. So bryophytes vaguely follow the Jepson ecoregions, as we've seen earlier, where the map that best represents or best compares with the Jepson ecoregions is the grid cell map, as I showed you earlier. And so from the Burge et al. 2016 study that I mentioned at the top of the presentation, this study showed that in terms of richness of species, the Northwest region had the highest species richness, followed by the Sierra Nevadas, followed by the Central Coast. But in our data set, and what we found is that it's quite different. The Sierra Nevadas is the most rich, followed by the Central Coast region, followed by the Northwest. And then lastly, our understanding of the bryophyte flora of California is still incomplete. We got to go out there and collect and make efforts to fill in the gaps in knowledge. Okay, so I think we talked about this in the beginning. I think we'll leave questions till the end. So if you have them, jot them down, put them in the chat, whatever's easiest. And so, okay, next to the next topic, floristics. So this is where, certainly the project I'm most passionate about. This was my master's thesis, and I'm still kind of wrapping it up. It's still being reviewed for publication. Hopefully I'll get it done by the end of the year. But this is a preliminary bryophyte flora of Henry W. Coe State Park in California. So a quick introduction to bryology in California, although you already got a good bit from the last presentation. I just want to go over some important notes here. So California bryophytes, they differ from much else of the state in terms of species composition because of the Mediterranean climate that California has, or that much of California has. And this figure here, this pie chart in a similar style to the ones we were looking at in the last presentation, shows the relative abundance of California's 10 most species-rich bryophyte families. The top 3 most species-rich families is Podiaceae, Gramiaceae, and Bryaceae. It's also important to note that, which is unique to California and to the West, is that compared to the eastern states in North America, the Western states have been, at least got, the Eastern states got a head start on the Western states in terms of studying bryophyte floristics. And we can see that in this figure here. Look at the documentation of North American endemic species through time. So in this, we can see from 1800 to 1900, we have these two lighter trend lines here. The gray one represents eastern species. This other one is a western species. And we can see that people studying eastern species were describing species at a higher rate from this in this time range here, but were quickly overtaken by researchers in the West. And this very bold trend line here is the average of the two trends from the eastern and western species through time. And over the last 50 years, we can see that this really steep upward trend from 1950 to 2000 is largely being driven by the discovery of endemic western species in California. So if one were to extrapolate and say if this trend continued, there's still many more species to be discovered in California. And of course, we can't conserve bryophytes without a baseline understanding of what is there. And so hopefully this study is a good example of how we can, one, fill in gaps in knowledge, and as well as lay the baseline for conservation. So now I can begin to hone in on the study site and where it is in California. So California has a number of mountain ranges, but today we'll be focusing on the coast ranges. So there's the northern coast ranges and there's the southern coast ranges. My study site is in the southern coast ranges. More specifically, this is called the Diablo Range, and it extends from Mount Diablo State Park in the North Bay, north of the Bay Area, all the way down south to San Luis Obispo County. So it's a very large mountain range. And so the Diablo Range is technically considered a interior coastal range because along these ranges we have an exterior range like the Santa Cruz Mountains and Big Sur, and we have this range separated by a valley known as the Diablo Range, an interior coast range. And the reason, and this range itself is actually really understudied for bryophytes compared to Santa Cruz Mountains and Big Sur and the more wet, more mesic areas. And that's one of the reasons why, is because bryologists historically are more interested in our abundant, more charismatic mosses that are found in more mesic sites like the redwoods. And so bryologists didn't come to this region to study bryophytes very often. Another reason it's understudied is because it's very rugged land, hard to reach. Very few people live in this area of the state, and it's mostly ranch land and private land, agricultural land, and there's only very few preserves for the public to visit. And so only one other study looked at bryophytes in this range, and that was Hutton et al. In 2021, where they did the Mount Diablo State Park bryophyte inventory. And that was done at the northern area section of the range. So focusing even further in on another subset of this range is the Mount Hamilton range. This is interesting and important to note for a few reasons. So Helen Sharsmith did a vascular flora of this range in 1945. Actually, she was the wife of the herbarium that I am currently the bryophyte curator at. And what she found was that in this range, just an hour and a half outside of San Jose and the Bay Area, lived a number of nearly endemic and rare vascular plants that which still hold up true to this day. But sadly, Helen did not look at any bryophytes. But anyhow, later on, in 2018, Kling et al. Looked at conservation priority of vascular plants in California. And this group looked at 3 different metrics: diversification, divergence, and survival. And will you know it, there is a hotspot for all 3 of these metrics right in Mount Hamilton Range. So for these 2 reasons and all the reasons I stated before, Henry W. Coe State Park makes a great study site for looking at bryophytes and for studying the diversity and the floristics in this area. So Henry W. Coe State Park is 87,000 acres large. It's the second largest state park in California behind Anza Borrego Desert State Park in San Diego. It spans from Santa Clara County to Stanislaus County. So like I said earlier, the climate of the park is largely Mediterranean, as is much of California. But this is certainly a more of a hot and drier Mediterranean variety than some other ones that you'd find closer to the coastline. The history and land use of the park is also quite interesting, and there are a few books on this if you find interest in this. But of course, we had indigenous people that roamed this park from originating both from the Central Valley and from the coast that used to forage here and hunt here. And some relics of that can be seen in terms of mortars and pestles. And then later on, Western settlers came. They put up ranches. They farmed. They planted orchard trees, as well as there's some mining operations that happened within the park. And then in 1935, the daughter of the very prominent Henry Willard Coe, rancher in this region of the state, donated the first plot of land to California, specifically to give those who live in the Bay Area an area of refuge and reflection from the busy city life. Okay, so I'm a bryologist, and I love flowering plants and I love our vascular plants, but I had to really focus on bryophytes in this study. So I made it really easy for me, and I kind of grouped the vegetation communities, the vascular plant communities, into three main groups that can be found within the park. And of course, there's great variety amongst these groups. But for me, and for the presentation, since we're focusing on bryophytes, I'll break it down to these three. So we have our foothill woodlands. So this includes a variety of different types of oaks and other trees. It can change from very open woodlands to very closed woodlands as they get closer to the riparian woodlands, which is another community type that is in the park. These can be deep canyons, or these can be more wet meadowy type ecosystems. And of course, we have plenty of chaparral as well in the state park. And these are also quite variable in terms of species composition of vascular plants, and cover certainly a large portion of the eastern side of the park where it gets more dry. The geology of the park is important to note as well. Most of it consists of a Franciscan complex where you find sandstone, conglomerate, blue schist, and chert minerals, largely the ones that you see in outcrops. There is a thin strip of ultramafic minerals in the park, and as we all may know, that serpentinite is a driver of diversity in vascular plants and occasionally in bryophytes. And so this area was certainly inventoried extensively to make sure we didn't miss anything. There are also a few volcanic substrates not shown on this map that are dotted throughout the park, and these areas were also surveyed, extensively. So like many areas in the West, these areas are home to wildfires. And so Henry Coe is no stranger to that. So over the last 20 years, there were two major wildfires that burned the park, one in 2007 and one more recently in 2020. So kind of right before I began my study. And so the most recent wildfire that hit the park was the SCU Lightning Complex fire. And this covered around 50,000 acres of land within Henry Coe. Some of it, kind of light fires, some of it more severe, where there were some tree die-offs. And you can see some of the scars of the fire in this image here to the left. But all in all, the park is recovering well, and is certainly is very good for many of the fire-following wildflowers in the park. And I think it's overall a good thing in the areas that wasn't so severe for the ecosystem at large. So back to bryophytes. So as I mentioned earlier, not many people have collected bryophytes in this Diablo range, let alone in the state park. So the first record of collecting bryophytes in the state park was Alan Whittemore in 1993. he and another colleague were planning to do what I did, was to create a flora or a checklist of the state park. But sadly, his colleague passed away, and the project was incomplete. And then from 1993 to 2020, various other people collected incidental collections within the park of bryophytes. And before I began my study, the total number of vouchers from the park for bryophytes was 186 vouchers, totaling in 90 species. So the objective of the study was basically to comprehensively inventory the bryophytes of Henry W. Coe State Park with the focus of diversity, distribution, and conservation implications. Although today I'm giving you somewhat of a shortened presentation of this study, and I'll really just be talking about the diversity of the bryophytes here in the state park. So over 3 years during my master's research, I took 32 field trips to the state park. So this varied by either walking in from the headquarters or other entrances by foot, backpacking, camping, driving my vehicle in, going off terrain, bringing other people with me, bryologists and vascular botanists with me to the park. It was a very, very enjoyable experience to go out to the park and see what was there. Not just looking at bryophytes, but looking at everything else as well. Once the bryophytes were collected, we processed them back at the lab. We put them in temporary packets in the field, and then once they're identified under the scopes, we put them in temporary or permanent packets. And those packets are now stored at the California Academy of Sciences herbarium. Also, all the records from this study are also publicly available on the Bryophyte portal website. And then duplicates from the study were sent to the UC Davis herbarium. And now to the results. The map on the right is a map of the state park. Pink dots represent the historical occurrences. So this is all the occurrences done from 1993 to 2020. And then the blue dots represent all the collections made during the study by myself and others. And so you can see from the map alone that we really tried to maximize coverage because it would be impossible to cover every single area, every single trail within the state park. It would take me years to do that. But we at least wanted to get a good range of east to west, north to south within the park, as well as go to places that were already collected from to confirm certain species were there or see if they had moved away or no longer there. So the collections to date after my research is 1,500 specimens. Species count has almost doubled to 179 species, which represents 22% of California's bryophyte flora. This included 91 county records from both Stanislaus and Santa Clara, so together, and then 3 tentatively undescribed species. So here's a nice part of the presentation where I get to show you nice, really pretty pictures that I took of bryophytes within the park. So feel free to just enjoy this part while I talk about mosses and things. So breaking it down by phylums, or by phyla, mosses make up 153 species within the park. And some of them you can see here from our proteaceous mosses that desiccate in the dry season. You can see in A and B, it's the same species, but it's just one's a desiccated version, one's a open version. From our bryogeous mosses, one of the more diverse families within the park. From Anicolia, which is this moss here with the lovely round capsules, as well as Aliconium androgynon here. And for the liverworts and the hornworts, so there's 20 liverwort species within the park and 4 hornwort species. Out of the liverworts, the family Ricciaceae, so all of your Riccias, it's going to be the most diverse within the park with around 7 species, and 3 of those you can see here. And then for the hornworts, there's only 4 species, but 4 may not seem like a lot, but 4 is actually half the total number of hornwort species known within California. And this includes 1 state endemic and recently described species, Theoceras prescauri, which the collection here in the park is an eastern range extension because this moss is really only known from the immediate coast ranges. So now we can break it down by family of the 3 most species-rich families within the park. So the first is Podiaceae. So these are some of our smallest mosses. These also include our annual species, which are hard to track down, especially during dry seasons. So last season was a really great season in 2023 because we got some really great rains and we added in a few new taxa to the list because, or at least I'm guessing or assuming because of the good rain season. You just get better collections of them and easier to identify. Also, you get better reproductive organs that you can dissect and use to key out. And so here are some of those species. Next is the Bryaceae family. So this family is interesting for a few reasons. So first of all, bryophytes aren't really known for their species that are introduced or invasive. It's something that's hard to study, but half of the introduced species within Henry Coe that we found are in this family and largely are native in Europe. Another interesting thing to note for Bryaceae is that many of the recently described mosses within this family over the last 10 to 20 years within, in California are actually super common within the state park, like this Gemmabryon binosum in the bottom right corner. And then all 3 of the tentatively undescribed species also are in this family, Bryaceae. Then the third most rich family within the state park is Brachythecaceae. No big surprises here. We have all the Scleropodium species, a few Homolotheciums, a few Brachytheciums. But the most uncommon out of the bunch, at least locally in the park, is Brachythecium frigidum, seen in picture A, which is really only found in some of the most wet and protected canyons within the park. So all in all, here's the species accumulation curve for the state park. We can see historically from 1993 to 2020, that's where that is. And then broken down by each year of collecting within the study, we see a still somewhat upward trend, which helps justify the title of preliminary flora of the park. So I imagine in years to come, maybe down the line, if more people get excited about this area, this region of the state and the country, people will come and collect at Henry Coe maybe on better rain years and maybe find some more interesting species to add to the list. So some specific noteworthy records within the park. Plagiobryoides vinosula in California is a special status species. It's CNPS rank 4.2, which means it's moderately threatened or endangered within the state. And this was found in both Stanislaus County and the Santa Clara County side of the park. And I believe it's just one of these mosses that is overlooked, and it's probably more common than it actually is thought out to be. Next moss is Alawina rigida. So this moss is technically new to California, although in the past there have been records of this moss, but these have since been annotated as they're misidentified as another common Aloina aloides, which is a more common species in the state. But now, so this represents the only record within California. And then, of course, the undescribed species in the Bryum family. We have the Plagiobryoides species, which can be seen here. This species is super common within the state park. I won't be talking about it in detail as someone else is already doing research on this, and it's a bit of a tricky situation. It's a species that's been known for a long time amongst bryologists within the West, but remains undescribed due to some taxonomic difficulties. But if you ever want to see it, it's easy to find if you ever visit the state park. It's found in the crevices of rocks within the creek systems there. Then you have 2 Bryum species 1 and Bryum species 2. I won't get into detail on Bryum species 2, but for Bryum species 1, I'll talk about in a moment. Here's a summary of the important bryophyte records from the state park. You can see the county records here, new to the Diablo Range as a whole, things that are rare from California, that is less than 10 occurrences from the state, introduced species, and then of course the listed species. So I want to mention this one species because I'm doing a little extra work with some other individuals. I'm trying to understand, well, trying to resolve the species in California. But here's a picture of it in the field, obviously very close up. But if anyone's familiar with this dichotomous complex of bryomes, you may know that it's really important to study the propagules of these plants, as they rarely reproduce sporophytes. In terms of the taxonomy, the propagules' shape, size, and morphology all are really important in getting to a species. And so this species in particular has very large propagules. And you could even call them deciduous branches, potentially. And you can see them circled in red here. They almost look like the normal main shoots of the moss, but they're actually tucked in the axils, the leaf axils of the moss. And we can see a comparison here between a more common Bryum gemilucens or Gemibryum gemilucens propagules that are found in its axils are very small, no leaves on them. And compared to this one, it has multiple leaves on it. It's basically already growing. And it's found in the axils of the leaves, like I said. So there's definitely a distinct taxa, especially here for California, although further work needs to be done to differentiate it from a morphologically similar species that's found in United Kingdom, and that species is called Bryum differenensis. So hopefully down the line, we can have better luck with resolving the species. Okay, so some main takeaways from this study. Again, I'll be driving this home throughout the presentation. Bryophytes are understudied organisms. We need to study them more. We need to get more people involved and excited about bryophytes, get out there and collecting. And of course, in California, relatively speaking, this is a very understudied region. And it's a large region that could be home to many more discoveries. Just in this one relatively small area within the range, we had all these interesting finds. And if we looked at some of the other preserves, so just south of Coe State Park, this is another really large preserve, BLM land. It's called San Benito. And certainly that would be a great candidate for another floristic study to kind of fill in these large gaps in this range. So then currently, Henry W. Coe State Park marks the most bryologically diverse region within the Diablo Range as of now. Okay, how are we doing on time here? All right, so the last topic, this will be a little bit shorter. This is something I've been working on more recently, and it's a little more fresh. But this topic has to do with speciation and taxonomy. So the title of this study is Morphological and Ecological Evidence for the Recognition of Anacolia baueri as distinct from Anacolia menziesii. So I'm working on this project with my advisor, Ben Carter. And so what got me interested in this project before I delve into it is that Anacolia menziesii and baueri is a species that I came across commonly within my floristic study of the state park. But it is a pain in the butt to identify, and it's a pain in the butt for all bryologists because it rarely produces capsules. And as far as we know, capsules is the only strong character in identifying these two species, or just distinguishing these two species from each other. So my effort in this was largely to try to find a gametophytic morphological feature new to this group that could help us differentiate these two species when we collect them without capsules. But there's a larger project to this as well. So Anacolia menziesii, Anacolia baueri, they span the west coast of North America into Oregon and into Washington, with this map here showing the blue in baueri and the menziesii being in red. It's a fairly common moss. It grows on vertical rock faces, occasionally on soil. And like I said earlier, it rarely produces capsules. But as we can see very superficially from this map, from this figure here, Anacolia baueri likes it a little bit drier than Anacolia menziesii, which likes it a little more wet, more mesic. And for this presentation, my advisor, Ben, is doing all the ecological and climatic analyses, so I won't be talking about that because we don't have that data yet. But this is just a precursor to that, where he's going to look at the morphological features and compare them to the distribution of these two species and the climatic differences. With that being said, Anacolia has a very complex taxonomic history. So first of all, the two taxa, Menziesii and Boweri, were described under different genera back in the 1800s. Eventually, the genus Anacolia was described, and these two taxa, Menziesii and Boweri, were put under this genus. Following this 1876 treatment of Anacolia, Flowers in 1935 did not agree that these two things were distinct species, and so included these things as distinct varieties of each other. So Anacolia menziesii, variety Menziesii, and variety Boweri. And so this treatment lasted for a long time. Someone else who did a treatment maybe in the 70s who included this as varieties as well. But it was not until 2004 that Norris and Shivak accepted that these two things are distinct as species. So now they're back. And also in their treatment, they were the first to note gametophytic differences between the two species. But then in 2007, when Griffin treated this genus in Flora of North America, he again rejected A. Boweri. So the dilemma is that people that are local to California who follow this Norris and Shivak treatment believe it to be a distinct species, but people that aren't from California or from the West typically don't accept them as unique species. So our goal here is to quantify the distinctiveness of A. menziesii and A. Boweri by demonstrating whether the characters above are significantly different. I'll show you those characters in a moment. And of course, determine the relationship between broad climatic patterns and these morphological characters. Like I said, we don't have those results just yet, but they'll be in the final publication. So here are the current distinctions between the two species or varieties, however you want to feel. So Menziesii has these round subglobose to ovoid capsules, as you can see in this image here. And Boweri has more cylindric oblong capsules. So these are the best. This is certainly the capsules is the best feature. Along with the capsules, another sporophyte feature is the spore size is larger in Menziesii, and then the seta length, which is the morphological feature that connects the capsule to the base of the shoot, is greater in Boweri than it is in Menziesii. And then the one gametophytic feature that Norris and Shivak included new in their treatment, which you'll see once I show you a picture, it'll make more sense. But it talks about the terminal subula divergence. So this basically is the shoots of the moss, whether the leaves, the terminal leaves are parallel or they're diverging or spreading. And you'll see a picture of that and it'll make more sense in a moment. So first, look at the results from the capsule morphology. So essentially, with all these features, I'll say that we took all these measurements using either calipers or using a micrometer under a microscope, and we took all this morphological data and we created these figures here. So for the capsules, we see that between the ratio of the length and the width, there are two very solid groupings or ranges in these ratios for Boweri and for Menziesii. And you can see those differences quite well in these dried capsules as well. And in this figure here, the groupings of the red Menziesii and Boweri in blue. There are a few outliers here, but like I said, the vast majority make them significantly different here. Now, the other two sporophyte features, surprisingly so, came out to be not significantly different. So remember, it was the spore size for the two species are supposed to be different, and the seta lengths are supposed to be different for the two species. But what we found when we measured these species is that, or these features, was that this did not hold up, and so will probably not be included in the next key or treatment of this group. All right, next is the gametophyte difference, looking at the terminal subula divergence. And so hopefully you can see it a little bit here. I know these are black and white photos, but you can see the leaf shoots for this Boweri here are oppressed, almost tulaceous, where the tips of the apices / subula of the leaves are almost parallel, where in Menziesii they are beginning to spread and they're diverging. And so this feature holds up certainly. As you can see from the box plot here, it is significantly different. There's a bit of range within A. Menziesii, but overall, most of the records we have here show a good split between the two ranges in the distance of divergence. Next, so this is the last feature, and this is the feature that we came up with that's new to this group that no one had pointed out previously. And so I was really excited when I found this, and we looked at the data, and we saw that there was a really significant difference. Let's look at the central strand diameter. So if you're unfamiliar with mosses and the terminology, totally understandable. But this is when you take a cross-section of a stem of a moss, you have different types of cells within the cross-section, within the cortex here. And so the centermost grouping of small, tiny cells is this called the central strand. And so we recorded using a micrometer the diameter of these of this grouping of cells, the central strand, for both species. And we found that that Boweri, Anacolia boweri, has a larger diameter compared to Anacolia menziesii, which has a smaller diameter. Okay, so here's a table showing the updated distinctions. So we got rid of the seed lengths, we got rid of the spore diameter, and we're left with 3 or 4 things now. The capsules are good distinguishing elements. The terminal subula divergence, the one I showed you previously, is different, and here the range is provided from 1 to 1.3 millimeters diverging, and the and 0.5 to 0.84 millimeters divergence. And the central strand diameter is different between the two species and have a really good separate range. So 35 and under micrometers for menziesii and 35 and greater for boweri. And then once the data for the ecological data and the climatic data comes in, it's very likely that it will show that menziesii prefers a mesic climate and boweri prefers a more arid climate. So what's left, like I said, Ben will do some of the ecological analyses, niche modeling. But in conclusion, morphological and ecological evidence does certainly support menziesii and boweri as distinct species. And this study is a good example of using quantitative approaches to bryophyte taxonomy. It should be pursued more in differentiating these cryptic species complexes. And it's definitely a good tool to use statistics in these instances to help differentiate these species. So some final words before I finish up and wrap up my presentation. I've been saying it this entire time, bryophytes are diverse. They are certainly understudied, and most of all, they are exciting, and they are a blast to work with. There's still so much to understand, and so much work to be done, and that's what makes me excited to work on bryophytes, is that there is no end in sight in terms of studies that we can do on bryophytes. Even if it's only just replicating what they've done on vascular plants, there's just so much to be done. Of course, I like to promote if anyone's really interested in bryophytes. I'm currently a part of the CNPS Bryophyte chapter, where we have yearly meetings in California and in other states. The next one is actually being hosted by your friend Jason Brooks in Arizona. And so we try to go throughout California for these yearly events and to other states occasionally. And so if you're interested at all, email me or get in contact with someone there. And it'd be a great place to get involved and to learn about bryophytes. And of course, thank you so much for having me here. I really enjoyed talking to everyone. I wish I could be there in person, but I'll take your questions now. Thank you.
Let's see here, hang on. Let me turn that. Okay, good. Thank you. That is a tremendous amount of work. Thank you so much, John. I had a question. Any questions in the room here? Is that about it? In the first part of your series, you were kind of showing the overlaps with the California floristic province and the species, and showing that the species were, and I can't remember, at the beginning I was a little distracted with the room setup, but that the species didn't exactly follow those California floristic provinces so much.
Mhm.
And I was wondering if you thought that it might be, and I'm thinking back to Jason's presentation and how much of the splitting of the species is happening around in our area. And I was
Absolutely.
thinking that as you're getting out into those different sub-provinces, if maybe what's happening is you're getting different, like what you've just done here with the, what was it, the Anicolia? Or
Yeah, totally.
yeah, that if we looked at them more closely, we might actually see that they're not the same species. We're, it's showing that it's going beyond, but maybe they're different species. That was what I was thinking. I just wondered what you thought of that.
No, that's totally a good point, and I think there could be a few theories on why, but that is very good, certainly a very good one. And I think it just happens to be, some, as you may know, taxonomists can be lumpers and splitters. And I think the splitting is definitely a more common trend in bryology these days. And people are beginning to realize with the help of phylogenetic work that bryophytes are more diverse than we thought, and that could definitely be leading to this kind of more fuzzy or fuzzy resolution for these different ecoregions that show more in vascular plants versus bryophytes. Maybe we're just not there yet. We don't see that distinctiveness yet. That's a great point.
Yeah, and years ago we lumped them a lot more, so that they were much considered to be really cosmopolitan, some of those species, but then looking at it more closely, it's not so much. I also thought that was really interesting that you were using statistics. I have a new favorite podcast called The Common Descent. It's all about paleontology, and that's what they do all the time, is they put measurements of bones, and then
Yeah.
they split out. Well, I mean, they also do genetics, but those measurements of the bone pieces is really how they'll get groupings of different things that to break them into species. I didn't realize that we weren't really doing that with living species.
Yeah, it's something that when I was learning about it too, I was talking to my advisor, Ben Carter, when I was doing this study and doing some other studies, talking about when you look into treatments and you see species, different ranges for different features, and he just says, when people describe those, they don't really, because I was like in a statistics class at the time, looking at like interquartile ranges and box plots. He's like, yeah, they don't really use that stuff. It's just kind of an estimate or just a range they came up with somewhere by looking at a few things. It's not like statistically a range. And so we're trying to, I think, why not use these like tried and true statistical methods and incorporate them in our living things? Makes sense to me.
Yeah, that's really interesting, because apparently it's like the method they really use with archaeology and things like that, paleontology. Yeah, good. Darlene has a question here.
Sure.
So not one.
Let's see if she can hear you.
DNA.
Yeah, not one mention of DNA, she says.
Yes, this is true. So for the last project, there was some genetic work done, but it was inconclusive. And so for now, I think we're going to go ahead to try to publish this. We have lots of good data, and when funding arises in the future, we'll hopefully to include the genetic portion then. But yes, it was done, and it just didn't help the cause. We need a bigger sample size. That's what we needed.
I can't remember, was Jason using DNA? I can't remember in Jason's presentation, and you might know, John, were they using, I remember him saying that you couldn't identify them in the field, you had to bring them home and look at them. It was leaf sections that I remember him talking about, but so I don't think he was doing a lot of DNA at any rate. Yeah.
I don't know what Jason was specifically what his talk was specifically about, but I'm not sure if he does DNA stuff.
I don't remember the topic, the name either, but it dealt with that kind of thing, and I know they were blowing apart species that surveyors have been calling the same thing around in here, but also saying that you couldn't identify them without looking at them. I mean, a lot of them, which is true anyway. But yeah, we need that. Isn't Brent Missioner, isn't he into the data swabbing idea?
Data swabbing.
Are you working with him so that you could just like do a swab of No, not data, I'm sorry, eDNA,
Oh,
eDNA kind of.
maybe. Yeah, he's into lots of stuff that I haven't caught up. He's
Okay.
leagues ahead of me. I mean, I'm sure he's
Okay.
been, but you're probably right.
Okay.
Um,
All right. Well, I forgot to mention in the beginning that we also have Meet a Plant, which is the really fun and interactive thing that we get in the room, and Frank Callahan is going to lead us in that. So you're welcome to go away, and folks in the Zoom also. You have to come up to SOU and join us to Meet a Plant in person. But thank you so much, John, for
Yes, of course.
presenting, and yeah.
I
All
had a
right.
blast. Thanks so much.
All
You
right.
have a
And
good
thanks,
night.
Willard, for arranging this. Yes. Okay.